All right, this lesson will be a supplement to Nancy Kanwisher's TED Talk about functionally specialized regions of the brain. You'll recall that one of the reasons that she and other colleagues suspected there might be a place in the brain that specifically processes faces is that there were some people with focal brain damage, so localized brain damage, who lost the ability to recognize faces. In fact, today we know that there are people who are born with this difficulty. So congenital face blindness is a condition from birth where individuals have difficulty perceiving, recognizing, remembering faces. So this is an example of how lesion data, so patients with damage to different parts of the brain, can reveal what different brain areas are doing. And lesion data is some of the oldest kinds of data about brain function, and it continues to be very useful today. Now, what you're looking at here is the surface of the brain, the cerebral cortex, the left hemisphere. Here's a real brain down here, so notice obviously there's no colors and there's no identifiable sort of regions like there are up here. But all these words here indicate a sort of a modern view of what different parts of the cortex are doing. For example, in the back here, it's labeled vision. And we know that this area seems to process vision in part because of lesion data. That's some of the oldest data. People with damage to the back part of the brain and the occipital lobe here are blind. They can be blind. So there are many ways to be blind. Some you can have problems with your eyes, but another way is to have damage to the posterior region of the brain here. Likewise, damage in the temporal lobe here can make people deaf, functionally deaf. Damage to this area here of the parietal lobe can make people unable to feel sensations on the surface of their skin. Damage to a motor region here can cause paralysis. Damage to these posterior areas here can interfere with language comprehension. In the frontal cortex, we have areas whose damage produces deficits in speech production, for example, and other lesions in the frontal cortex interfere with other kinds of cognitive tasks. So the idea then is that for a long time, lesion data helped to map out the functional architecture of the brain. So now with the invention of the fMRI technique, researchers can examine an intact brain, right, in a healthy brain, and also folks with brain damage, but they can measure activity in a healthy brain when the subject is doing different kinds of cognitive tasks. And here she showed her brain on successive days as she was looking at faces or objects. And the fMRI machine was measuring relative brain activity all over the brain when she was looking at faces or objects, and found this little spot right here on the underside of the right hemisphere. It looks like the left here, but we're looking from underneath the brain. This area here showed specifically increased activity for faces over objects. Now, just to get a better sense of where that is, if we take a look at this picture here, we're looking at the brain from the underside, right? So this is the bottom of the temporal lobe, here's the bottom of the frontal lobes and the cerebellum and so on. So this would be the right side, because if you turn this brain over, then this would be the right hemisphere and this would be the left hemisphere. So we're looking at the fusiform gyrus down in here, right? And so the face area is a localized patch of brain tissue right there. Okay. Now, what kinds of stimuli was she presented with? Well, all kinds of different faces. And notice these faces, they have a lot of different sort of low-level visual characteristics. They look different. I mean, here's a dog's face, right? Here were some of the object pictures. So a part of the body, typewriter, hand, and a scene here. And these two are interesting. These are Mooney faces, and so you might not have recognized it initially, but that is a face. And this one is inverted. And again, that little spot of tissue, the fusiform face area, shows significantly increased activity for all these types of stimuli relative to these types of stimuli. Now, in addition to the fMRI data, which is measuring the functional activity in an intact brain, I wanted to introduce another kind of data that can help us understand what different brain regions are doing. And here we're going to look at single cell recording in the brain. So brains are made of brain cells called neurons, and researchers can make very, very small electrodes and insert them into different regions of the brain to record the electrical activity of individual neurons. You can also use these electrodes to stimulate areas of the brain. But for now, we're just going to think about an experiment that involved recording from cells in a certain region of the brain. In fact, what these researchers did in monkeys now, so this will be a monkey brain, is they used fMRI to identify that cortical patch of tissue in the right hemisphere that processes faces. So they had the monkey passively looking at different objects and faces and scenes. All the while, the fMRI machine was measuring brain activity. And sure enough, they localized the region of brain that selectively, preferentially responded to faces. It's the very same one. It's the equivalent to the spot in Nancy Kanwisher's brain. So this will be the fusiform face area in the monkey. Now, after they found that area, then with the fMRI, then they went ahead and they inserted microelectrodes into that region of the brain, that region of the cortex, to record from individual brain cells what kind of stimulus was its preferred stimulus. We can see down here the general idea. So we have the firing rate. This would be a measure of the electrical activity of that individual neuron when the monkey is looking at different objects. So for non-preferred stimuli like scenes or objects, you'll get low-level activity. But for the preferred stimulus, in this case faces, you get significant increase in activity. Right, and so the fact that these brain cells are showing a preference for faces is why the whole region shows a preference for faces. And we're going to define this idea of a preferred stimulus as that kind of stimulus, in this case faces, that generates a maximum response. Now, you never know if there's some other kind of stimulus that could generate an even higher response, but studies have been done where they showed thousands of different kinds of stimuli. And these areas in this area of the brain respond maximally to faces in humans and in monkeys. Now, the actual results of that study, when researchers inserted electrodes, they recorded from a whole bunch of cells in that general area, and they found about 97% of the visually responsive cells in that area of the brain, the fusiform face area, were strongly face selective. So that fits with, it dovetails nicely with the fMRI data, that the reason why there is a region of the brain that responds in the fMRI selectively to faces is because there's a whole bunch of brain cells in that region that are selective for faces. Well, while the previous team was recording from cells in the cortex from monkeys while they presented different stimuli, another kind of data that reveals the function of a brain area is stimulation data. And you saw kanwisher played that clip of this guy here who has epilepsy and was about to undergo surgery to have a bit of his brain removed, the part that was causing the epilepsy. Oftentimes, doctors will implant electrodes to record, to identify which part of the brain is triggering, is causing the epilepsy. And it's just so happened that some of the electrodes were in the fusiform face area, so they wanted to see what would happen if they stimulated that fusiform face area. And sure enough, when they stimulated it while he was looking at the doctor's face, it produced perceptual disturbances. He described it as sort of the doctor's face sort of transformed, maybe into somebody maybe he'd seen before. A very weird experience. And again, that supports this idea that little bit of tissue that Nancy identified in the fMRI and that has many face-selective cells in monkeys, if you stimulate that bit of brain, you interfere with face processing. So in her TED Talk, then, Kanwisher goes on to identify other bits of brain that seem specialized to process different types of stimuli. So we have the face area, and this, by the way, is an inflated view of the brain, right? We have regions that process color and scenes in the green here, and there's a whole bunch of different little bits, um, places in the pink, the faces in the blue, visual words. Notice that's fusiform, but in the left hemisphere. So the face area is predominantly on the right side, the right hemisphere. The left hemisphere fusiform gyrus seems to be processing visual words, the form of words in your known language. There are regions that become active when people are thinking about other people's thoughts, looking at bodies and biological motion, etc. In fact, then towards the end, she mentions these areas here in white, and these areas become active when people are doing thinking, when they're manipulating information. Maybe you're doing mental math, or you're thinking about generating a map in your mind and how to go from point A to point B. When you're manipulating information or holding information in what psychologists call working memory, when you're utilizing information and transforming it, these areas become active. So Kanwisher's arguing that the brain does seem to have a set of functionally specialized regions. Now, how they come to be that way is a matter of ongoing research. But fMRI and lesion data and stimulation data are filling in this picture of what different parts of the cerebral cortex are doing.